Okay, I'm going to go ahead and get started. So today the talk is called ProImmune T cell proliferation assays, and I'm going to be going over our suite of assays that are based on an ultra-sensitive CFSE flow-based method and designed to assess functional T cell responses to any antigen. So just a little bit of background. So my name is Andrew, and I've been with ProImmune now for about half a decade. My own education was more focused on post-translational modifications of protein, but I've been working on these types of immunogenicity risk assessment projects now for quite some time. So what do I mean by functional T cell assays? So these are the gold standard for understanding potential antigenicity of a peptide or a protein of interest. These can be used to assess both wanted or unwanted immune responses. So wanted in the case of something like vaccine development, or unwanted in the case of probably most biotherapeutics that we're talking about. Unwanted immune responses can result in delaying in development. They can cause loss of efficacy of your drug. They can cause altered PK and PD. They can activate unwanted B cell responses that can lead to downstream effects like anti-drug antibodies, and ultimately things that will cost you development time and money. So the important thing to keep in mind is that this is applicable for the development of any biologic, really, ranging from things like antibody-based drugs, ADCs, bispecifics, through the development of newer things like generic peptides, immune modulators, engineered proteins, things with new junction regions, looking at epitopes or any kind of immune therapy targets, vaccine development, mRNA candidates, you name it. I listed most of those things here, but pretty much the list goes on. And so the assays that we have cover a few ranges of applications, and kind of plug into your pipeline a little bit differently depending on where you are in development, and depending on what type of candidates you're developing. So we offer the ProMap T cell proliferation assay. This assay is a way of measuring relative antigenicity. It doesn't use any other cell types. It's just using healthy donor PBMCs from a primary source. The This assay does allow for T cell epitope resolution. So the source of this assay is peptides, and so because of that, you have an individual peptide that's giving you a response or not, and so you're able to kind of narrow down exactly where you might be seeing a functional response that way. And nicely, because of that, we offer synthetic peptide synthesis. So if you're early in your development, you can get away with doing this with nothing but sequence in hand. The other assays that we offer are the ProScern DC-T, and also our Mixed Lymphocyte DC-T. And so both of those require our monocyte-derived dendritic cells, and they require mixing of cell types. So in the case of the ProScern dendritic cell proliferation assay, we would be remixing the monocyte-derived dendritic cells presenting your protein antigen with autologous PBMCs from the same donor. In the case of the mixed DCT lymphocyte reaction, we would be taking cells that are from monocyte-derived dendritic cells from donor 1, and we would be mixing those with PBMCs or T cells from donor 2. And that causes a mixed condition, which drives an allogeneic response, and we can look for immune modulatory responses. All of these assays require our ProImmune PBMC biobank. And so this biobank is sourced from altruistic donors in the UK. We are provided with the blood for PBMC isolation. We do all of the cell isolation and cryopreservation ourselves. So we have a lot of control over the quality of these cells, and reproducibility of these cells as well. And these are all fully HLA type for MHC class 2. This gives us a really good way of looking at specific populations or just generally tailoring it to a global population. I did see a question pop up in the chat. I'll get to any kind of questions. If you do have anything, definitely feel free to put them in there, and I'll just circle back at the end and answer any questions that I haven't resolved. So the first assay that I wanted to go into is our ProMap T cell proliferation assay. And as I mentioned, this uses either peptides generated from overlapping peptides. This can be overlapping from an entire antigen source, for example, or from an entire engineered region of an antigen. Or they can be peptides that were identified using a MAPS assay, like our ProPresent, which is another assay that we offer. But they could also be in silico predicted or just the peptide that you're studying itself in the case of a generic peptide or a process impurity. And so we would take CD8 positive depleted cells, PBMCs from these healthy donors. A typical project for this ranges from about 20 to 50 donors. And then we would generate overlapping the peptides of interest for it, and we would incubate those for 7 days. The controls for this include our tuberculin PPD, which is a whole protein antigen that all these donors have been exposed to at some point in the form of vaccine. And so it's a very potent recall antigen. But we also offer keyhole limpet hemocyanin, which is a very potent naive antigen source, as well as CEFT, which is a cocktail of peptides from CMV, EBV, flu, and tetanus. So these are very strong recall antigen peptides put together into a peptide pool. And we also offer a KLH peptide pool. This is generated from peptides that were identified from keyhole limpet hemocyanin, but they're just individual peptide epitopes. And so again, this proliferation is measured over 7 days. We're using an ultra-sensitive CFSE method to do this. So all of the PBMCs are dyed using CFSE. And then as each cell is divides, you get half as much CFSE in each subsequent division. And so over time, the cells will kind of move over into this dye negative area. And these are indicative of highly proliferative cells. And so just diving into that a little bit deeper. So this is what it looks like when you have an unstimulated control. And so you have on the y-axis, you have your CD4 response, and on this x-axis, you have your CFSE dye. And so these cells in the B quadrant are high dye positive CD4 T cells. And again, as they proliferate, you're getting fewer and fewer dye, less dye in each proliferative event. And so those move over here, and these represents the proliferated cells. And so what we're able to do is look at percent proliferation as a function of the what comes over here in quadrant C over the total population of C and D together. And so the percent proliferation cells are just compared to the percent background. And if this is above a p-value of 0.05 by ANOVA, these are considered a significant response. And that, together with the average strength of the response, gives us what we call a response index. And so the response index is a function of both the percent donors that are responding, but also how strongly those donors respond. So it takes into account both factors. You wouldn't want to sway the results by a large number of donors that are responding to a very low level antigenicity versus something where just a few donors are responding very heavily. Those types of things weighed out. And so this is what the data would look like for this kind of a project. You have all of your different peptides along the x-axis, and then you have the different donors stacked up on the y-axis, leading up to a total of 100% if you have all the donors responding. And so for something like PPD or KLH, we tend to see 100% responses. That's very typical. These are kind of nuclear bombs. Most people tend to respond to them. For something like the CEFT or KLH peptide pools, you still get very robust responses as well. And then when you look at typical peptides I think this is for an antibody drug, you would see responses, in the fewer sporadic range. And so the way we tend to interpret this is that if you only have one or two donors that are responding, it's a bit of a, you want to pay attention to it, but it could be kind of attributed to a sporadic response. By the time you're getting 3 to 5 donors responding to something, it's a bit of a yellow flag. And if you're getting more than that, it's definitely something you want to look at. And so a good talk that kind of drives into this exact approach is on our website by Dr. Tim Hickling. And so this is data that he had at his time at Pfizer. He's over now at Roche. And what he did when he was working at Pfizer was they applied this to their antibody pipeline development. And what they did was look at a similar kind of traffic light approach where anything that only triggered one or two kind of got filed under a green light. More than that, it kind of got under a yellow light, and then at a certain cutoff they applied, they filed it into a red category. And so this is a good example of how you could apply this kind of data. This is in the development of bocukizumab, a very famous antibody. And they looked at in silico prediction of this, and they found 3 potential epitopes across the CDRH2, CDRL2, and CDRL3 regions. And then when they dove down and actually did the functional assay, it turns out only one of those was a high-risk sequence. And so this is a good example of how in silico is useful, but at the end of the day, depending on how you weight your assumptions in the in silico work, you really can kind of either over or under predict things. Another good example of how this could be used is this was also provided on our website as a talk by Professor Toru Okumaru at Osaka University. And they were developing an engineered ACE2 decoy as a way of neutralizing SARS-CoV-2 infection. And they were looking at a number of different ACE2 candidates based on their binding affinity, but they also wanted to look at potential immunogenicity as a way of ranking these out. And so looking at the ProMap data, they identified a sequence, L79F, that stimulated multiple donors. And so they decided to swap that out with the T92Q sequence, which was shown to have a lower risk for immunogenicity here. And so the next assay I want to talk about has a similar approach in the sense that it's still using a CFSE-based flow cytometry readout as a way of measuring T cell proliferation. But this is using now a different approach where we would have monocyte-derived dendritic cells do natural antigen processing and presentation, and then they would turn around and use those same autologous cells with their own PBMCs to see if they're able to drive a T cell response. And so a typical donor panel for this is in the similar range of 20 to 50 donors. Again, these are all fully typed for HLA class 2, so we are able to match specific populations of choice, or we can also just kind of look at global coverage for that. And then the dendritic cells are generated from monocytes. Then they're loaded with your protein of interest. They're allowed to process and present, at which point we wash them and then co-incubate them with unmanipulated CFSE labeled PBMCs. This goes for 7 days, just like the ProMap assay, and at which point we would look for proliferation by flow. There is a limited exposure of test articles, so we do avoid unwanted assay interference by doing a number of wash steps. But it is something to always keep in mind that if you have a very strongly activating T cell, directly activating therapeutic, for instance, there might be some direct effect that you can never eliminate completely. And so a good example of how this works is this data provided to us from Elasmogen, and they were looking at a number of Shark VNAIR domains, BB10, 2G, BA11, and EO6, as compared to the control KLH and tuberculin PPD proteins. And so, again, these are off the charts in terms of their response index. Pretty much everybody will respond like that. But if you look at their 4 candidates, all things considered, you would go with something like BB10, which shows a lower risk of immunogenicity, especially compared to something like the 2G candidate. Another good kind of example of how this could be used comes to us from Eisai, and this is also available on our website. Dr. Peng Li spoke about how they applied this to their antibody development pipeline as well. They put all 11 antibodies that they had into a panel for DC-T, and they were able to then get kind of a really nice sense of relative risk assessment that way between all 11 candidates. And so the last assay I have to talk about this afternoon is our mixed DC-T lymphocyte reaction. So our MLR assay, this is a really nice way of looking at immune modulation. And so it's a little bit different in terms of the data that you get back. Rather than kind of measuring relative antigenicity of a protein, now you're able to look at the effects on the actual immune modulation that it might have. And so this uses a similar monocyte-derived dendritic cell, but instead of incubating it with autologous PBMCs, we would incubate it with allogeneic PBMCs from a different donor. And so that gives you the mixed lymphocyte condition in the name. And so a good example of how this looks for an autologous condition in an unstimulated control, almost all of your CD4 cells are unproliferated. When you provide an antigen source, such as a protein, an antibody, something like that, you'll get some degree of proliferation, as seen here in quadrant C for the stimulated variety. For the allogeneic condition, however, you end up seeing around 60 to 80% of your cells in a stimulated condition. And so the mixed lymphocyte condition drives about 60 to 80% proliferation. And so what this allows for is anything that would increase or decrease that effect can be seen as, and you can kind of get that immune modulatory effect in either direction. So an example study for this, we have 3 donors providing 6 donor pairs. And so these are all labeled as D1, D2, and D3. And so you would use either the monocyte-derived dendritic cells or the T cells from any given donor, and apply all possible pair conditions. And so for just a given pairing, this is an example for donor pairing 1, you would have CFSE labeled responder cells from donor 1. You would have your unlabeled dendritic cells. These are the presenter cells from donor 2. And then you would have them mixed as a mixed condition. And so you would have 6 different versions of this. And then along with that, you would incubate your test conditions. And so in this case, we have your controls as well as your drug product in 5 different concentrations. Sorry. And so each data point here represents the average mean stimulation of 8 replicate wells, and each donor would be done in triplicate. And so a typical non-drug control condition, you see about in this case, it's about 55, but about 60 to 80% is typical for the mean stimulation here. And then when the drug is applied at the highest concentration, you can see that there's an immune stimulatory effect. And that's titrated out as you kind of decrease dosage here. And so that's kind of a really nice way of seeing if you have immune stimulatory or immunorepressive effects of protein, peptide, small molecule, whatever drug you might be looking at. So to summarize all this, T-cell assays really are the best solution for immunogenicity risk assessment, and they're quite an important part for developing your biologic pipeline. T-cells can be challenging to set up. In order to get good data out of them, you really have to have high-quality cells. You have to have a diverse set of HLA types. You have to have stringent protocols in place to get reproducibility. And we offer a very high level of expertise in all this. Our flow-based method offers a high level of sensitivity to detect low precursor frequencies. So this can be used quite nicely for naive antigens as well as recall antigens. And it's also a really good way of getting this data very fast. So we offer the fastest turnaround in the industry of about 3 to 6 weeks from when we get your contract and your test articles in hand. That's when you would get your data back. And so we do offer a very timely turnaround. And we act as partners in this process to provide quotes, a study design, and any kind of other information you might need about assessing the immunogenicity for this. And together, this is offered as part of our integrated suite of assays. And so we do offer tools. I think I had already mentioned the MAPS assay. That we call that ProPresent. It's an antigen presentation assay tool. But we can also do Reveal, which is an in vitro MHC peptide binding assay, as well as ProStorm, which is our innate whole blood assay looking for innate immunogenicity. In addition to our services, we also offer a line of products. This includes our new Anchoron high affinity protein binders. We're really excited about these. Definitely come and speak to us about that, and our MHC multimer platform as well. And so ProImmune, we're located in Oxford. We've been doing this for about 25 years. We're experts in this field. We have global reach now. We're headquartered in Oxford, but we also have personnel in the USA and in Singapore that cover the rest of the globe. We're ISO accredited, and we have customers in over 50 countries. These customers include publicly disclosed relations with the FDA, a number of large academic groups and pharma groups, as well as smaller biotechs. And with that, I'd be happy to answer any questions. I know there was a few in the chat. Just taking, yeah, Sophia, I know you had one.
Yeah, I was just asking if you guys ever tried to do a recall with the peptide in the T-cell proliferation assay, if the response is higher after, giving another dose of the peptide?
So that's a good question. So for the same donor cohort, probably not. Generally, the assay sensitivity is such that it is very good for naive cells, and it would absolutely work for a recall response if it existed. But because these are healthy donor PBMCs, that unless it was something they would naturally encounter, that's why we use the CMV, EBV, flu, and tetanus pool. But I don't know that we've done kind of a recall experiment on that same donor cohort, that kind of thing. I would have to ask about number of cells. There'd be some considerations to think about.
Thank you.
And in terms of peptide testing concentration, so I don't want to misspeak. I believe it's 4 micromolar, but we do have it might be 5 micromolar, but we do have a consistent empirically derived peptide testing condition that we found has been kind of reliable for looking at these naive responses. Are there any other questions? Okay. Well, if there aren't any other questions that do come up, please do feel free to reach out to us. We would put you in touch with a team member who can get you any quotes or answer any questions that you might need. And my own information, I'm Andrew. You can always find me online at the ProImmune website. And definitely feel free to send any of your questions our way.
Thank you so much.
Thank you. Appreciate it.